Thank you so much. This is my first time attending this meeting, and I'm so motivated, inspired by the work that everyone is doing. And one of the benefits of going after Paul and others who presented is that most of the foundation has already been laid. So I think I'll be able to spend a lot of time talking about, unfortunately, when things don't go so well in gene therapy and how we move forward in that case. So I want to talk about Skysono, which is the FDA-approved gene therapy for adrenoleukodystrophy. And as we've been talking about study design and things, I think it's helpful to see sort of how we got to this point of having the FDA-approved product. And as this is one of the early ex vivo gene therapy products approved. And when we went to the FDA very early in the process, we would say things that, ALD does not get graft versus host disease, and they're like, prove it. And so you had to have that as your primary safety outcome. And you had to go back, and a lot of work was done by Paul and others in this room to look back and find natural history. So the foundation of the approval comes from two studies that were retrospective, so looking at the natural history of ALD, or retrospective and partially prospective, sort of a natural history of boys treated with allogeneic stem cell transplant. And then two foundational studies, so ALD 102 and ALD 104, which were two prospective gene therapy trials, non-randomized, no placebo, just patients who were getting this on an open label trial. And I'll talk mostly about those two studies. And as we all know, the fifth trial that's been involved, or study, I should say, is a long-term follow-up. The FDA for ex vivo trials requires 15 years of follow-up. And one of the things that we can also talk about probably for hours in a panel if we wanted to, was that there's no mechanism for that. There's no funding for that. And so if you're doing gene therapy outside of industry, it's really hard to find out that model for doing appropriate long-term follow-up. But we have that ongoing. And as I'm going to talk about, it's incredibly important. Um, I'm going to breeze through a little bit of this because we did hear about most of it from Paul and others today. But for patients with cerebral ALD, there are two cellular therapies currently available. So allotransplant, where, as you know, the donor is another person. Patients get prepared with myeloablative chemotherapy. Then the stem cells are infused. For gene therapy, it's a very similar process, except for the patient is their own donor, just like we were just hearing about from Dr. Orchard. Those patient has their stem cells collected after 5 days of getting the same medication we were just talking about, gene therapy transduction in a lab, and then go through myeloablative chemotherapy. And then the goal or the expectation is that a portion of those stem cells that are derived from either the donor or the genetically modified cells from the recipient migrate into the central nervous system and can differentiate into uh, cerebral microglia expressing the ALD protein. If this slide looks very familiar, it's because Dr. Orchard just presented this almost exact slide. So I will just go through this to say ALD gene therapy and MLD gene therapy are both ex vivo products where those stem cells are collected and infused later. We have a long-term follow-up study. The original trial that we're going to talk about, the two trials were 2 years in duration with a long-term follow-up extension for a total of 15 years. So the two studies that primarily got us to approval were the 102 and the 104. ALD 102 was opened between 2013 and 2018 and enrolled 32 boys. The second study, the follow-up study, was 2019 through 2021 and enrolled 35 boys. So just the key enrollment criteria, boys had to be less than or equal to 17 years old, so children or young adults with active cerebral ALD, defined as having an MRI less score between 0.5 and 9 with contrast enhancement present, and neurologic function scores of less than or equal to 1. So very similar to what are often used in transplant, although there are transplanted patients who go older than that, more advanced than that. In the first study, because allogeneic stem cell transplant was a standard of care at the time, you could not have a match-related donor transplant. Things were going so well after the first one, we said, families should have a choice. And so if you have a matched sibling and you wanted to have transplant, that's okay. The key efficacy outcomes were many things that you'd expect. So patients who are alive and free of major functional disabilities at month 24. We looked at changes in less score and the NFS score, resolution of contrast enhancement, and then the vector copy number in the peripheral blood. And then safety outcomes looking at neutrophil engraftment. So did your stem cells grow after you received the product, adverse events, and others. So this is the kind of Kaplan-Meier overall survival curve that we all want to see. And so this shows you ALD 104 and 102, those two studies of 67 boys, and the overall survival. It's hard to get much better than that, but as we all know, overall survival is not everything. And so then we look at the major functional disability-free survival, and it still looks pretty good. It's not perfect, but still looking good. So overall survival in red, and the major functional disability-free survival in blue. So still doing well. Um, and again, major functional disability is a relatively low bar. And so what we also looked at was the physical functioning, school functioning, emotional functioning, and psychosocial health summaries or scores of patients over time. And what we see is that there are some dips at different points within the period, within the course, especially in emotional functioning, but there is no significant difference between the initial scores and the last. So patients go through a lot, but ultimately get to the same point. And it would be great if we could say that things were better, but that's just not, that's not what we expect after transplant and not what we see with gene therapy. Um, we want to know if this is a durable response. And so this looks at the vector copy number or the copies of the copies per diploid genome, excuse me, of the patient to see if these are stable. And so in the red boxes, these are patients who had a higher vector copy number when the cells were infused, and they are stable at a higher level than the blue, but those are all appropriate acceptable levels over time. So indicating durability of response, in this case, of longest follow-up up to 108 months, although with our first patient treated in 2013, we're getting further now. And this is now the challenge. And so I think what Dr. Orchard was telling us is similar about ALD and MLD, that allogeneic stem cell transplant compared to gene therapy is a lot easier in the early course. And so there was no transplant-related mortality in the patients treated on the studies, but there's a transplant-related mortality of up to 18% in the allo transplant, no graft versus host disease. You're getting your own cells. You're not going to have graft versus host disease, compared to around the 35% in the allotransplant population. No graft failure in the gene therapy compared to 17% in the treatment population. And what I mean by that are patients who would have met eligibility for the gene therapy trial were they to be enrolled, compared to the entire matched population of allos. And so then the grade 3 treatment-related toxicities, very similar between these, and they are all consistent with getting chemotherapy. Here is the problem, and probably where we should spend most of our time. There are 10 cases of myelodysplastic syndrome, or AML, which are blood cancers, reported in the patients treated with gene therapy and none in allotransplant. So those Kaplan-Meier curves don't look so great anymore when we look at event-free survival. So the event-free survival, with events being defined as death, need for a second transplant, graft failure, or major functional disability, we see ALD 104, and that's the second study, having a much worse outcome or higher, much more inferior event-free survival compared to the first study, and also then you see those sort of in the middle compared. And the data that I'm presenting is from our New England Journal paper last year, so it only has 7 patients, and we are now up to 10 patients since that time. I know not everyone in the room is an oncologist, so just to talk about MDS and AML, they're basically blood cancers that perform along a spectrum, where you start with a normal production of blood, myelodysplasia, you end up seeing some changes in the bone marrows and lower blood counts over time, and left untreated, this progresses to a frank AML. And so AML being a very serious leukemia. And what is challenging for all of us is that the treatment for MDS in children is an allogeneic stem cell transplant. So basically what you're trying to avoid in the first place. We've spent a lot of time trying to determine what was different about those boys who developed cancer compared to those who did not, and lots of exhaustive detail trying to figure it out. The most obvious, although probably not the entire solution, was the boys who had a higher risk of cancer actually had a lower intensity of chemotherapy. So chemotherapy called busulfan and fludarabine, compared to the boys in the earlier study who had a higher intensity busulfan and cyclophosphamide. I think it's also worth noting that every other gene therapy, or I think every other gene therapy, uses busulfan alone. In this trial, used a combination of chemotherapy for historic reasons. And so to say, how did these boys get treated? All of the boys who've developed cancers to this point have gotten an allogeneic transplant. We have the published outcomes for 7 of those boys. One child died of complications of his allotransplant after gene therapy. The others are alive. One of them has relapse MDS and is having a second transplant. And then it's too early to know the outcomes for the other three, although they are alive. So why did this happen? Just to say that the purpose of a vector or the integration in ex vivo gene therapy is to put the gene somewhere. For ALD, we know that this is an X-linked disease, but we don't put the gene there necessarily. It goes anywhere. And so when you have that vector or that virus carrying the gene to a spot, it's going to land somewhere. And the problem with this is where it's landed in many cases, or that integration site from where the protein is made. So I know I'm tight, so I'll move quickly. So inserted genes, they influence the activity of the things around them. So if you put a gene near other genes, a lot of times it doesn't do anything. It doesn't change them. Sometimes it can make those genes make more of their protein, sometimes less, and other times lead to uncontrolled growth. The problem is when you insert a gene near a gene that can increase the risk of cancer, either by downregulating it in something called a tumor suppressor gene or upregulating it in oncogene, that can lead to cancer. And that is what's happened in each of these boys. And so the problem, we believe, or at least part of the problem, is how this very specific gene vector was designed. It's so the backbone is an HIV-derived vector that gets rid of the parts that make you infected, but takes advantage of the fact that HIV puts DNA material into cells. This was paired with an MMD promoter, so the piece in the red, that was linked with the ABCD1 DNA. And that MMD3 promoter is a very strong promoter that drives a lot of expression. And that is exactly what we think the challenge was. That promoter was driving expression into a specific gene, something called the MECOM locus, where all of the boys treated with gene therapy. So those diamonds on the bottom that just look like a black mess, those are every boy on that study has integrations in MECOM, every single one, but 10 of them have cancer. And so we're trying to determine why. We know why MECOM causes or increases the risk, but we don't know why some boys develop this and others don't. And so the obvious question is why was it designed like this? Why was this done? So the reasoning, the rationale goes back multifold. So it wasn't known when the trial was designed how much expression we needed to drive. And we know that MNDU3 promoter drives expression. And people felt strongly that we have one shot at this for boys to preserve neurologic function. So try to drive as much expression as you can. This was not the first trial of gene therapy for ALD. The first trial was done in France using the same vector, and there were no cases of leukemia in that study. The study went through every single preclinical safety test that existed and passed all of them. So did not identify oncogenic risk, and it's used in other diseases like immune deficiencies. All of that said, this does appear to be something very specific to this product. The company Bluebird Bio, who makes Skysona, also does gene therapy for blood disorders of sickle cell and thalassemia. So the big rectangle on the side has all of the integration sites, and the most common integration sites for people treated for sickle and thal, compared to the small rectangles on the other side. Those are the boys with ALD. That MECOM locus doesn't appear anywhere in the other patients who don't have ALD. So it's something very specific to this product. So what happened, I think you know this part of the story. LE-Cell, which is now Skysona, received approval from the FDA Advisory Committee in June 2022. At that time, there were 3 known cases of MDS. The FDA predicted 11 based on the preclinical work. There was full approval in September 2022, and it's currently offered at 5 sites. I know that I'm up on my time, so I'm just going to skip over this next part quickly, just to say where are we going from now, or where are we going? So there are multiple different academic centers and companies who are looking at different gene insertion techniques, so using different vectors, not using that promoter to decrease the risk. Gene editing, so very directly correcting the ABCD1 mutation. In vivo therapies that we heard a lot about earlier. And there's a company out in San Diego that's looking at, there's a company out in San Diego that's looking at glial repair. But I think many people have heard the recent story about personalized editing that came out of work done at Children's Hospital of Philadelphia. So all of these are areas that we're looking at. And maybe just my last slide, and I'll skip over the rest, just to say what genome editing is. So that shotgun approach where the gene gets placed anywhere, that is what we see with the type of vector we're using. Genome editing targets a very specific spot in the DNA sequence, uses an enzyme to clip that spot, and then specifically repairs the ABCD1 gene by either inserting, deleting, or changing it. There are challenges. It's a little less efficient than insertion. It can't be done with every single mutation, and it can be done, though, without viruses, which is great. So we've heard a lot about in vivo therapy, and these are things that are being worked on. There are challenges with the delivery to the brain and off-target effects, and so we're still working on that piece. Personalized gene therapy is actually being done, not for ALD yet, but there's conversations about that being done at a group out of Berkeley and other places. So this might be coming, though it is very cost intensive and labor intensive. So in conclusion, I think hope through science is really important. So despite all of this, I do think that Skysona gene therapy is a valuable treatment option for some patients with cerebral ALD, particularly if you do not have a matched donor. It has a potential to halt disease progression. We saw that, and to improve outcomes. You don't have to find a donor, and not everyone has a donor. You don't need to have those risks of graft versus host disease. But the risk of insertional oncogenesis is real, and we need to think about it very carefully. My hope is that emerging therapies will offer more alternatives, including the editing, personalized therapy, and others. And so with that, these are some of the many, many patients who helped us get to that point. And just want to also then acknowledge all of my collaborators, many of whom are in this room. Thank you.